viewers, in my last lecture on nucleic acids, I had described the sugar and the nitrogenous base components of nucleic acids. While both purines and pyrimidines are heterocyclic aromatic compounds, yet they can be distinguished from each other based on their chemical structures. A purine has 2 carbon rings, whereas a pyrimidine has 1 carbon ring. The purine has a pyrimidine ring fused to an imidazole ring. The pyrimidine has only 1 pyrimidine ring. Thus, we can say that purines have 4 nitrogen atoms, whereas the pyrimidines have 2. This you can see on your screens. On your screens, I have showed you the pyrimidine ring, and what we find that pyrimidine ring is a heterocyclic 6-membered aromatic ring. It has a basic nucleus of pyrimidine, and we have 2 nitrogen atoms which are forming the part of the ring. Hence, it is known as heterocyclic ring. And then you can observe that the numbering of the ring is done in such a manner so that the nitrogens get the lowest possible number, and the numbering is done in clockwise manner. Whereas when we look at the purine ring system, we find that it has 2 rings in it. One is 6-membered ring, like this is your pyrimidine ring, and in this the other one is the imidazole ring, and both these rings are fused at 4th and 5th carbon. And what we find again here, that the numbering is done in such a manner so that the nitrogen atoms get the lowest possible number. What you observe here is that a pyrimidine ring has 2 nitrogen atoms in it, which are at position 1 and 3, whereas a purine ring has 4 nitrogen atoms in it, which are at 1, 3, 7 and 9 positions. So what you have to remember, that pyrimidine ring is a single ring. It is only made up of pyrimidine, and this pyrimidine ring is going to be the basic structure in all the pyrimidines. Hence, we can say that pyrimidines are derivative of this pyrimidine. Whereas, when we'll talk of purines, they will have this basic nucleus, and then all other purines will be derivatives of this nucleus. We'll modify it. Now we know that purines include adenine and guanine, whereas pyrimidines include cytosine, thymine and uracil. So again, here I've shown you the structures of these, the basic structures, the ring structures, which are present in purines. And what we find, that in purines we have double ring structures, that is, we have 2 purines are guanine and adenine. And what we find that both of these purines, whether it is a guanine or adenine, both of them are present in DNA and RNA. When we say DNA, what we mean here is that the basic sugar in DNA is deoxyribose sugar, whereas in RNA, the basic sugar is the ribose sugar. And the 3 pyrimidines that we have are cytosine, thymine and uracil. And what you find here again, that this is a single ring structure. Now what we know further, that in cytosine and thymine is present in DNA, whereas cytosine and uracil is present in RNA. These 5 nitrogenous bases, which are purines and pyrimidines, are regarded as primary bases since they are fundamental units of genetic code. The nitrogenous base or nucleobase that makes up the nucleic acid are used to distinguish DNA from RNA. So that means we can distinguish between the DNA and RNA on the basis of the sugars which are present in them. And the second thing on basis of which we can distinguish between the two are the bases, which are also known as nucleobases, which are present in them. Because in DNA, thymine pairs with adenine, that is whereas in RNA, it is uracil which matches with adenine. So where does the difference lie? The difference lies that in DNA, thymine is there, which is a purine, whereas in RNA, the purine is uracil, whereas in both the pyrimidines are adenine. Now, how does thymine and uracil differ? They differ by having a methyl group. That is, in thymine, we have a methyl group which is absent in uracil. So what, again on your screens, I have shown you the structure of thymine and uracil, and what we find that in both these pyrimidines, the basic structure is the same. So where is the difference here? The difference is that in thymine, we have a methyl group, and in uracil, this methyl group is different. So where do DNA and RNA differ? The DNA and RNA differ in their pyrimidines, whereas in both of them, the purines will be same. That is, in both of them, what we find, the purines is adenine, whereas in both of them, the pyrimidine is different. That is, in DNA, the pyrimidine is thymine, whereas in RNA, the pyrimidine is uracil. Now, the pairing of nucleobases, that is, cytosine with guanine and adenine with thymine in DNA, or adenine with uracil in RNA, are referred to as base complements, which we shall be studying later in this lecture series. Let us now shift our attention to nucleosides and nucleotides. But before we do that, let us quickly recapitulate that what are the units which will be present in DNA and RNA. We are going to have the sugars, which can be a deoxyribose sugar in DNA or a ribose sugar in RNA, and then we are going to have bases which are purines and pyrimidines. These are going to be the basic units in nucleosides and nucleotides. But nucleotides will be different from nucleosides in the sense a phosphoric group will be present, a phosphate group will be present in nucleotides, which will be absent in nucleosides. So let us discuss now nucleosides and nucleotides in great detail. So what are nucleosides? Nucleosides are compounds in which nitrogenous bases, as I have said, which may be purines and pyrimidines, they are conjugated to a pentose sugar. Now this pentose sugar may be a ribose sugar or a deoxyribose sugar, and these bases are linked to a ribose sugar or a deoxyribose sugar by a beta glycosidic linkage. Now that means what you have to remember is that the C1, this I will be showing you with the structures also, that the C1 of the furanous form of the ribose or deoxyribose will be linked to the nitrogen of the base. Hence, these are known as beta and glycosidic linkage. Now, according to the classical structural definition, we can define a nucleoside is an N-glycoside of D-ribofuranase or D2-deoxy-D-ribofuranase. Now, what do we mean by this? We mean that a glycosidic linkage is formed between the first carbon of the ribofuranase form of ribose or deoxyribofuranase form of deoxyribose, and the bond formation is taking place between the first carbon of either of the sugars with the nitrogen of the base. Hence, it is known as N-glycosidic linkage. Now, initially it was considered that nucleosides are compounds of ribose or deoxyribose with a pyrimidine or a purine. But as the time passed, this definition was extended to apply to purines and pyrimidine and glycosides of almost any carbohydrate. So now this becomes a very vast definition in which it takes up all other carbohydrates also. The purine or pyrimidine part of a nucleoside is often called as a purine or pyrimidine base, or it can also be called as purine or pyrimidine nucleobases. Now, the main difference between the D-ribose sugars and 2-deoxy-D-ribose sugar is that when we talk of RNA, we have a hydroxyl group at 2' position. Whereas when we talk of deoxyribose sugar, there we have hydrogen in presence instead of an hydroxyl group. Therefore, this sugar is known as 2-deoxy-D-ribose. 2-deoxy means that the second position we do not have an hydrogen atom. And this difference in the structure, that is the absence of oxygen at the second position of deoxyribose sugar, greatly affects the structure and stability of DNA and RNA macromolecules. So now let us look into the structure of the furanase form of D-ribose and 2-deoxyribose. Well, as I've said that when we look into the structure, we'll start doing the numbering from here, and when we are talking of DNA and RNA, these numbers will be indicated with prime, that is 1 prime, 2 prime, 3 prime, 4 prime. That is in order to distinguish the numbering between the carbohydrate moiety and the nucleobase. Now, what we find when we are saying ribose, what we mean here is that it is indicated in yellow, that at second position you have a hydroxyl group. Whereas when we talk of deoxyribose, what we find that we have only hydrogen here. So oxygen has been removed. And since this oxygen has been removed from the second carbon, it is known as 2-deoxyribose. To differentiate or to stress on the point that oxygen has been removed from the second carbon and not from third or the fifth carbon or the first carbon. So this is what we mean. And when we say the structure is furanase structure, what we mean here is that it has a 5-membered ring, that is the basic structure in furane, and O's indicates that it is a carbohydrate, that is it is a sugar. Now, if we look into the structure, I wanted to explain two things. We are referring again and again to capital D. What do we mean by saying capital D? If you look into this is the open chain structure of ribose. And when we look into this structure, what we find that the OH will be on this is the structure in which the OH is on the right hand side. So this is referred to as D form. Now, when we make a bond here what do we find? That the bond is being formed between the OH on the fourth carbon and the carbon on the first atom, resulting in a hemiacetal. When this bond is formed, a new chiral center is generated here. And what we find that at this center, when the bond formation takes place, that is, we are forming a intramolecular reaction is taking place, resulting in the formation of hemiacetal. Now two configurations are possible. One is the OH is on the upper side of the plane, that is the CH2OH and OH are on the same side. If such is the case, then this molecule is known to have beta configuration, and we call it as beta D ribofuranase. Whereas if this OH was below the plane, then we would have called it as the alpha form. So what you have to remember, that is, when we are talking of DNA and RNA, there the ribose and the deoxyribose have configuration beta at the anomeric carbon. And I know that you are familiar that anomeric carbon is the carbon which is linked to two oxygens. So that is how you are going to distinguish. And this is the first carbon, that is, this is the aldehydic carbon, which has been converted into a hydroxyl group here. And this is this hydroxyl oxygen which is being converted into a ether group here. So this is the basic structure of a ribose or a deoxyribose if we remove oxygen from here, and the configuration is going to be beta because OH on the first carbon and OH on the fifth carbon are on the same side of the plane. Now the conformation here may be syn or anti. Now because sugars have so many hydroxyl groups in them, it is because of this that these sugars will make nucleosides more soluble in water than if they were free bases. Now what you have to remember is, that is, what does the B-glycosidic linkage involves? The B-beta glycosidic linkage involves the C1 prime of sugar and the hydrogen atom of N9 in case of purine, or N1 in case of pyrimidine, thus eliminating a molecule of water. That means this is a condensation reaction between the OH group of the C1 prime of the sugar molecule and hydrogen at the ninth position, N9, of the purine, or N1 in the pyrimidine, eliminating a water molecule and resulting in the formation of N-beta glycosidic linkage. So as you can see on your screens now, this is the purine base. And when we talk of this purine base, what we find that the linkage is taking place between the N9, that is, hydrogen will be removed from here and OH will be removed from 1 prime of the ribose sugar, right? OH and H will be eliminated and a bond will be formed here. Now, as I've been saying again, that is, I'll repeat that the bond is between the beta OH group, that is, this molecule has beta configuration here, that is, we are calling it as beta glycosidic linkage, and the bond is between carbon and nitrogen, therefore we call it as N-beta glycosidic linkage. And the other thing that you have to take note here, that in the ribose, it is the numbering has been done as 1 prime, 2 prime, 3 prime, 4 prime and 5 prime, whereas in a base, numbering is simply 1, 2, 3, 4, 5, 6, 7, 8, 9. So you have to remember, if the bond, this is universal, between a sugar, ribose and a purine, then the bond will be between C1 and C9. And if it is a pyrimidine, then the bond will be C1 and N1. of the base. Now here again I have shown you the structure of a purine, and which is of a nucleotide, and in this case the base is adenosine. So what do we have? And the sugar here is deoxy sugar. So we've named it as 2-deoxyadenosine. This is the IUPAC name, and the bond is between the C1 of the deoxyribose sugar and N9 of the purine, which is your adenine. So this is named as 2-deoxyadenosine. If we talk of the pyrimidines, again what we observe here, that the bond is between the C1 dash of the deoxy sugar and between the N1 of the pyrimidine, which is cytosine here, and we get a beta N glycosidic linkage, and the name of the molecule or the nucleoside is 2-deoxycytidine. I'll be discussing that why is it cytidine shortly when we discuss the nomenclature of these molecules. Now when we look at what we conclude from here, that since in purine, when we are taking purine as the base, the bond is between C1 and N9 of the purine. Therefore, purine nucleosides are N9 glycosides, and the pyrimidine nucleosides are N1 glycosides. Because see, in pyrimidine, the bond is going to be between the N1 and the ribose sugar, and whereas when we talk of the purine, the bond is going to be between N9 and the ribose or the deoxyribose sugars. So purines are known as N9 glycosides and pyrimidines are known as N1 glycosides. Now what you have to remember next is that if a nucleoside is having ribose as a sugar, we will call it as ribonucleosides, whereas the nucleosides which have deoxyribose as a sugar, we will call them as deoxyribonucleosides. Now the second thing you have to remember is that nucleosides are generally named after a particular purine or a pyrimidine present in it. So that is, if that is the case, how will we derive to the name of the nucleoside? What we'll do is, we will add idine to the root name of pyrimidine and ocene to the root name of purine. So let us see how we will do it. So if the purine is adenine, what will we do? We will convert ene into ocene. That means adenine will become adenosine. That is the name of the nucleoside. If it is guanine, which is again a purine, so what will you do? You will convert ine into ocene. So the name will become guanosine. If it is cytosine, which is a pyrimidine, you will convert this sine into dine, that is ocene into dine. So the name will become cytidine, whereas with thymine it will become thymidine. For uracil it will be uridine. So this is how we are going to name the nucleosides. So what are you going to remember? Adenine will become adenosine, guanine will become guanosine, cytosine will become cytidine, thymine will become thymidine, uracil will become uridine, and the conformation here again can be syn or nt. So now on your screens what I have done, I am sharing with you the base. If the base is say adenine and the sugar is ribose. Now we are talking of the systematic name of the molecule. So this will become adenosine ribonucleoside, and the common name will become adenosine and the abbreviation will be AR. Similarly, if it is cytosine and the sugar is ribose, the name will become cytosine ribonucleoside. And you know the common name will be cytosine, cytidine, and here the name will be CR, that is the abbreviation. Similarly, we can do for the sugar. When the sugar is deoxyribose, it is here adenine, so it will become adenine deoxyribonucleoside. The name will here become, the common name will be deoxyadenosine, and the name will be ADR. Here ADR is indicating that the base is adenine and the sugar is the deoxy. This D is indicating that the sugar is deoxyribose sugar. So here I have shown you the structures of a ribose nucleoside, and what we find here is that the bond is again between the C1 of the sugar and N9 of adenine, so the name becomes adenosine. That is, this is a purine. Whereas when we look into a pyrimidine, again here the sugar is ribose and the bond is between the C1 dash of the sugar and N1 of the pyrimidine, which is here cytosine. So here the name will become, the nucleotide will be named as cytidine, and here it will be named as adenosine. And you have to remember that these are the common names, these are not the IUPAC names. Similarly, if the sugar is a deoxy sugar, so the name will become deoxyadenosine. Here D is indicating that the sugar is deoxyribose sugar, and adenosine is indicating, adeno is indicating that the base, nucleobase present here is adenine, and the name of the nucleoside is deoxyadenosine. Similar is the case, that is, we've taken an example of deoxycytidine. In this case, the sugar is deoxy sugar, ribose sugar, which is indicated by C and D, and C is indicating that the base present here is cytosine and the nucleoside is named as deoxycytidine. So if I summarize this, so we can say, quickly summarize this, that a nucleoside consists of a nitrogenous base linked by a glycosidic bond to a C1 of a ribose or a deoxyribose. The nucleosides containing ribose are named as ribonucleosides, while those possessing deoxyribose are named as deoxyribonucleosides. The nucleosides are generally, please remember, named after particular purine or pyrimidine present in it, and then you have to remember that if it is a pyrimidine, we will add idine to the root name, that is the base name, that is, and if it is a purine, we will add ocene to it. So that means adenine will become adenosine, that is an example of purine, and if we take an example of pyrimidine, which is thymine, which is present both in DNA and RNA, it will become thymidine. So for this lecture, I had referred to these references, that is by Robert, that is concepts of genetics by Nelson et al., by Pierce et al., Klug et al., and Garrett et al. These are the books which I had referred to. In my next lecture, now I will be taking you on to a journey to study as to what are nucleotides and what is the difference between a nucleoside and a nucleotide. Thank you.